Okay, so I can start. So hello again. My name is Joseph Farah, Microfluidic Application Specialist at Fluigent, and I'm delighted to guide you through today's session. We are honored today to have with us Dr. Dan Cojoc, Senior Scientist from the Institute of Materials from the National Research Council of Italy. Dr. Cojoc has extensive experience in optics, nanoscience, cell biomechanics, developing advanced experimental systems that combine microfluidics, optical tools, and cell mechanics research. Today's webinar will be structured in several parts. So the first, we will start with a small introduction about cell mechanics and the micropipette aspirations technique. Afterwards, we'll dive into the application with Dr. Dan about the red blood cells mechanics and mechanosensitive ion channel activation. And finally, a Q&A session where you can ask your questions directly. So please feel free to use the Q&A box at the end, at the bottom of the Zoom part at any time, and we will address your questions after the main presentation. Before we dive into the technique, let's first understand why cell mechanics is fundamental for studying how cells respond to force and maintain their functions. Okay. So cell mechanics, why it matters. So cells constantly experience mechanical force in their environment. For example, the red blood cells in circulations are usually exposed to hydrostatic pressure, forces from fluid to pressure differences, shear forces from the flow, from blood movement and fluid flow, tensile forces stretching within the cell membrane, and compression forces, which are like squeezing through narrow spaces of tissue constraint. These forces influence critical processes, including like the cell shape and their deformability, mechanotransduction, how cells convert mechanical stimuli into biomechanical signal, and cell migrations, differentiations, and disease progression. Changes in these mechanical properties of the cells often indicates disease. For instance, red blood cells can become stiffer in sickle cell anemia or malaria, while cancer cells often become softer than their healthy counterparts. Studying these changes requires precise quantitative methods capable of probing cells mechanics at a single cell level. So one of the most used and established techniques in this area for this purpose is the micropipette aspiration. The principle is simple, yet it can be powerful. So basically, as you can see in the image, there is like a glass micropipette with a micron scale tip. It is brought into contact with the cell membrane. This membrane afterwards, so we can have a controlled suction pressure where it is applied, and part of the cell is drawn into the pipette. By varying the pressure, we can measure how much the cell can deform and extract parameters. This technique, as you can see, is not invasive. It can keep cell viable and provide real-time reproducible measurements of single cell mechanics. However, to unlock the advantages of these techniques, the micropipette aspiration usually is based on the total pressure at the pipette tip that comes from two components. So the first one is the hydrostatic pressure, which is set by the height of the liquid reservoirs, providing a stable baseline for the flow, and the pressure controller, or the microfluidic pump, which can fine-tune the pressure applied to the cell for suction or pushing. The pressure difference, as you can see in the schematics on the right, the pressure difference that is stable, the pressure difference between the inside and the outside of the pipettes can determine the cell state. So for example, if it is stable, like delta P, it is 0, like you will have the baseline. If it is aspirated, that will have a negative pressure, and a push out if it is a positive pressure. Accurate control of this difference ensure the quantitative, reproducible, and reproducible experiments using these techniques. So to achieve this precise control in the micropipette aspiration, high-performance pressure systems are essential. So that's why the Fluigent pressure controller can offer a high resolution for very small pressure adjustments with an excellent stability and repeatability, ensuring reproducible results, automated software control for complex protocol, and these features make the experiments, as mentioned, more quantitative and can be consistent. So one of the why we can choose the Fluigent, the pressure controller compared to the other systems. So the Fluigent pressure controller is particularly well suited for the micropipette aspiration, since it can combine precision, stability, and speed with key advantages offered traditional syringe pumps. So for example, you will have a pulseless flow that provides continuous stable pressure without any oscillations compared to the syringe pumps, on which already have, for example, some mechanical force, which can induce some oscillations, as you can see on this graph, and can provide, the Fluigent can provide like faster times in terms of the response, which guarantees a better control over the micropipette technique. So fast response time down to 30 milliseconds, allowing instant pressure changes for dynamic experiment, a resolution down to 0.03 full scale %, a stability of 0.1 and an accuracy of 0.25 with a high repeatability with 0.01%, less than 0.1% of changes as the full scale. This combination makes the Fluigent ideal for studying, for example, red blood cells mechanics and mechanosensitive ion channels activations with high precisions and reproducibility. Within this expertise, today we are pleased to introduce Dr. Dan Cojoc, senior scientist at CNR-IOM in Italy, with over than 30 years of experience in optics, nanoscience, and biophotonics. Dr. Cojoc had developed more than 20 optical tweezer systems, advanced microscopy and spectroscopy platforms with over 150 plus papers published, along with multiple patents. His current research focus on red blood cell mechanics and mechanosensitive ion channels activation using one of the techniques, the micropipette aspiration. So today we are honored to have you, Dr. Dan Cojoc, and we will be pleased to hear and see your explanations about this topic.
Okay, thank you. I now share the presentation. I guess.
That's perfect.
Okay. So, hello everyone. My name is Dan Cojoc, and I am with the National Research Council of Italy, Materials Foundry Institute in Trieste. And today I will speak about how we use the microaspiration technique to activate piezo channel in a red blood cell and correlate this with mechanical properties and recent applications to Alzheimer's disease. Just to have a look where we are. So we are at the northeast of Italy, close to Croatia and Slovenia. Here you can see the area science park, where we have the electrosynchrotron and Fermi-free electron laser. So it's a large area science park, and our institute is over here. And my lab, called the optical manipulation, because it's based on mainly on optical tweezers microscopy, is within this institute. The outline of my presentation is as follows. So first I will mention briefly mechanical properties, deformability mainly of the red blood cells. Then how the alterations of red blood cell mechanics can influence disease, and how we can use this alteration as diagnostic biomarkers. Then how the red blood cell volume and mechanical stability is regulated by piezo channel. Then about the technical approach that we apply to measure red blood cell mechanics in concert with piezo-1 activation, so in the same time. And I will show that this red blood cell mechanics and piezo-1 activation are correlated. Then I will discuss about the recent application showing that alteration of the piezo-1 activity in red blood cells of patients with Alzheimer's disease is, the alteration of the disease alters the red blood cells in terms of piezo-1 activation. And finally, the conclusions. So just a few words about the red blood cells. They are present in the blood of 41%. The plasma is 55%, and the other components like platelets, white blood cells are at a low percentage. Probably all of you know that the main function of red blood cell is to carry oxygen from the lungs and deliver it throughout our body and transport carbon dioxide back to the lungs. This occurs by the binding of oxygen molecules to hemoglobin, and then these molecules, oxygen molecules, are released in the capillaries near the tissues because of the pH change, but also because of the mechanical constraints. Just some numbers and facts for who doesn't know. First, the number of red blood cells in our body is very high. So they are about 10 trillions, representing about 83% of the total number of cells in the human body. They are produced by bone marrow by a process called erythropoiesis at a rate of 2.4 millions per second. The lifespan of a red blood cell is about 120 days, during which it performs about 100,000 circulation cycles. You can imagine how many deformations should such a cell perform in this so many cycles. Red blood cells exhibit remarkable deformability, enabling them to adapt their shape and navigate through arteries, veins, splenic sinuses, and blood capillaries, which are smaller than their size. So deformability is essential for the cell. Some morphological and mechanical properties. From the morphological point of view, the discocyte shape maximizes the surface area, about 40% more surface area than that of a sphere with the same volume. The shape, still the shape, gives a momentum of inertia, which is bigger than that of a sphere. This makes the red blood cell less prone to rotation during flow. So it avoids to rotate, and the circulation is better. Biconcavity facilitates capacity to deform via high surface area to volume ratio. As we know, red blood cells lack nucleus and other specific organelles present in a typical cell. Then the cytosolic hemoglobin concentration dictates intracellular viscosity. So mainly hemoglobin, which is the major element inside the red blood cell, dictates the viscosity. And the viscoelastic membrane comprised of lipid bilayer and flexible membrane skeleton primarily determines deformability. And now, to measure morphology and mechanical properties, a useful technique, and I think the eldest technique in mechanobiology for cells, I would say, was introduced by Hochmuth in 1986, I think. This is a review. And the technique consists in capturing a cell with a micropipette and then increasing the pressure, making a tether of the cell membrane enter into the pipette and measure the size of the tether. So we can measure the size of the tether inside, we can measure the radius of the cell outside the pipette during the aspiration, and using some physics, Laplace law, we can determine some nice properties of the cells like Young modulus, membrane tension, cell viscosity, bending modulus, and hemoglobin concentration. So these are typical values measured by micropipette aspiration for red blood cells, leukocytes, with, by micropipette aspiration, as I said. There are, of course, also other techniques that allow to measure mechanical properties of the cells. Among these, optical tweezers is one technique that I use a lot. The difference is that in this case, the forces which can be applied are much, much smaller. So it is a difference in the range of forces. Atomic force, microscopy, microfluidic devices, microfiltration, and ectacytometry. So each of these techniques have their advantages and disadvantages, as all the techniques that we use in our research. But the differences mainly consist in the range of forces that they can be applied, the throughput, and the possibility to couple them with other techniques of observation. Now, just few properties of micropipette aspiration. While you can see a micropipette and which takes from the solution red blood cells, the first red blood cell is aspired and then released and then aspired again, just to show that we have a good control with the pressure on that. Then we increase the pressure and we can suck, let us say, many other cells. This technique is easy and low-cost implementation. It has a relatively good throughput for single-cell techniques, so we still do single-cell experiments, but with a good throughput. And the mechanical stimulus is in the physiological range for the cells, and it is relatively easy to couple with fluorescence, which allows to investigate mechanosensitive channels activation, as you will see in a moment. Here is schematically, we show the alterations of red blood cell morphology and mechanics in disease. So you see a series of disease, sickle cell disease, hereditary spherocytosis, sepsis, inflammation, malaria, diabetes, thalassemia. All of them, as a consequence, they deform the morphology and the mechanics and alter the mechanics of the red blood cells. I do not enter into detail for each of them here, but just giving an example for malaria. So the parasite remodels the red blood cells and the ring forms are visible on smear. The stiffness of the cell is increased, the deformability is decreased, inducing sequestration. Or for instance, sickle cell disease, hemoglobin sickle polymerization induces stiff crescent shape of the red blood cell. So as a consequence, lower deformability and higher fragility. Now, one can use this alteration of red blood cell mechanics as disease biomarkers. For instance, deformability, which can be measured also with micropipette aspiration, can be used as biomarkers in sickle cell disease, in malaria, and diabetes. Membrane rigidity or elasticity, again, which can be measured also by micropipette aspiration, can be used in hereditary spherocytosis as biomarker, I mean, thalassemia and sepsis. also another mechanical property is cytoplasmic viscosity, still another addition to endothelium, and fragility lysis threshold. Now, we spoke about the technique in general. We spoke that we said that with this technique we can measure the mechanical properties. What about measuring the activation of mechanosensitive channels in the membrane of the red blood cell and in other cell also? Piezo1 is a mechanosensitive channel which is an integral membrane, so an intermembrane protein, which plays a crucial role in fast signaling during mechanosensory transduction process in almost all living cells. The one nice, interesting characteristic about piezo1 in red blood cell is the fact that in red blood cell, the piezo1 is the only mechanosensitive channel. So it allows to, in this case, we have the chance to study really the properties of the piezo1, not together with other families of mechanosensitive channels. So mechanosensitive channels are what they are. They are proteins, integral membrane proteins, activated by mechanical stimuli exerted on cellular membranes, upon which they rapidly and efficiently convert these stimuli into electrical, osmotic, chemical, intracellular signals. They have been shown to play a key role in many physiological processes associated with mechanosensory transduction, including hearing, touch, proprioception, and blood flow regulation in mammalian cells. So they are quite important to guarantee a good function of the cells. And what it is interesting is that the piezo1 and piezo2 mechanosensitive channels, they were discovered relatively late in 2010 by Ardem Pataputian and David Julius. And they received the Nobel Prize in Physiology or Medicine in 2021 for their discoveries of receptors for temperature and touch. So these are senses. The touch is a very old sense that we always had, but the receptor, so the molecule which is responsible of this, was discovered just 15 years ago. Piezo1 is a structure, as you see, so there are 3 blades here and the cap and 3 beams integrated into membrane. And what is a relatively big protein, 200 nanometer per 10 nanometer. And when the membrane is tensed, the blade are expanded and this channel is open and the cap goes a bit up, letting the ions go from outside to inside the cell. So it's a mechanical activation of the protein, of the channel, to let cations, as you see here, non-selective channel, let them go inside the cell and signal to the cell different stuff, like here. So in red blood cell, the volume of red blood cell is regulated by signaling through piezo1. As you can see here, figure out, you see the piezo. Due to the microcirculation membrane stretch, calcium ions enter into the cell, and this calcium activate another channel, the potassium channel, and this makes, and also the aquaporins, this makes water be released from the cell outside the cell. So, and the volume of the cell will change. And this is necessary when the cell travels through the body. No? Piezo 1 and calcium influx through piezo 1 can be also associated to the dynamic cytoskeleton reorganization and ATP. Okay, I realized that my mouse was on the other monitor. Now it should be on the right monitor. So, as I said, contributes to dynamic cytoskeletal reorganization and ATP release through PANX1, Panexin 1. Okay, so we have cytoskeleton reorganization and the release of ATP through this channel. But this is signaled by the calcium ions through the mechanosensitive channel. Then if there are mutations like loss of function or gain of function, these are manifested on overhydrated red blood cells or dehydrated red blood cells, which is found in hereditary xerocytosis associated with resistance to malaria. Even without mutations, mechanical, metabolic, osmotic, or pharmacological factors can alter piezo 1 functions in red blood cell. These effects contribute to red blood cell dehydration, reduce deformability, and shorten lifespan, playing a role in conditions such as sepsis, hemolysis, or in mechanical devices like ECMO and valves, and red blood cell storage lesions. Okay, so how we can investigate the piezo 1 activation by mechanical stimuli, again with using micropipette aspiration on red blood cells loaded with fluorescent dye, fluo4 for instance. And as you can see in the sequence of images, when we increase the pressure and the cell deforms more and more, at a moment we see the fluorescent signal, which means that the channels open and the calcium ions enter from outside the solution into the cell. Now, how we implemented this technique in our lab, as you see in the scheme of the experimental setup, we have an inverted microscope. Here is the sample with red blood cells. We illuminate in bright fields the samples to follow them when they are sucked in the pipette. And we block this filter yellow. It is very important because we block the wavelengths which were responsible with the fluorescence excitation. And on the other hand, here we excite for fluorescence, okay? And we collect both signals and we split them on two different sensors, but the recording is occurs simultaneously, concurrently. And this is, in our opinion, big advantage because we can study both mechanics and piezo 1 activation for single cells in the same time. In the image here, you see Hamamatsu camera, the microscope, and the Fluigent pressure device. Okay, so now the technical approach that we use in this setup is illustrated in this movie. So you see the red blood cell. At the moment you will see it, that it is attached to the top of the micropipette and then sucked in size. And look at the fluorescence image. Okay, the fluorescence increases at the moment. And then we continue increasing the pressure and the fluorescence decreases because the fluorophore bleaches, and also we lose the information because the membrane is destroyed. So what we have a relation between the pressure. Okay, we increase gradually the pressure. We measure the deformation of the cells, that is a tether inside the pipette, and we measure the fluorescence signal, and we define the activation as the moment in which the fluorescence signal begins to rise up, to grow very fast, as you can see around the point P. What we have before, we have here, for instance, in this step, we have the contact between the cell and the pipette. So first is a contact, and then we have a higher pressure and activation of the channel. Okay, now what we studied the correlation between mechanics and piezo 1 activation, because we had the chance to have these values together, okay, together for the single cell. So we could study a correlation. And first we induce an actin instability by treating human red blood cell with cytocalazine at low concentration, measure the mechanical parameters and the pressure for piezo 1 activation. Then we compare them with normal red blood cells. The treatment with cytocalazine takes effect after 23 minutes. So we expect that the cells treated with the drug do not change the properties before 30 minutes, but they do change the properties after 30 minutes. Studying was made possible because our setup allowed to measure them concurrently for every single cell. Excuse me if I mentioned more times this, but it is the main novelty as a technique that we introduced, and we think that it is important to mention. The experiments address the following questions. Does actin instability affect cells' deformability and mechanical properties, such as Young modulus, membrane tension, and viscosity? Does actin instability influence piezo activation? And finally, is there a correlation between mechanical properties and piezo 1 activation? Now, here are the results for control cells and for treated cells. They were considered quite big numbers of cells, more than 250 cells. Just to show you, so these columns refer to the time period, so from 0 to 20 minutes, 20 minutes to 40 minutes, and 40 minutes to 60 minutes. What we can see that indeed the values of the pressure for piezo 1 activation are lower for treated red blood cell after 20 minutes. The values of the mechanical parameters are also lower as an effect of the induced actin instability. To show better this, I captured the main values. So you see the values in the first interval at the beginning. There are not big differences between control and treated cells. But when we go to 40, 60 minutes after treatment, we have indeed big differences in terms of mean, median, and pressure for 50% activation probability. Then we studied the correlation between the activation pressure with Young modulus and membrane tension. And to see if the values are relevant, we did the following. We arranged and paired the data for pressure, activation pressure, and one mechanical parameter randomly. So those are the R here. What we observe again, that for after 40 minutes, there is a strong correlation both for control and cyto for the cells we measured. If we mix the cells, so we take cells or we take the pressure activation randomly and randomly from mechanical properties and couple them, then there is no correlation. So this indicates that indeed at the single cell level, we have a correlation between mechanics and piezo 1 activation, both for control and cyto. Okay, and finally, the application or our experiment with red blood cells, which were treated with beta amyloid characteristics, fragment proteins characteristic for Alzheimer disease. Sorry. Just some introduction about Alzheimer disease. Probably you know, is a biological process that begins with the appearance of a buildup of proteins in the form of amyloid plaques, as you can see here in this figure, and neurofibrillary tangles in the brain. The tangles are figured out here. I hope they are observable. This causes brain cells to die over time and the brain to shrink, as is illustrated here. So it is a shrinking of the brain as a consequence of this alteration of the cells. Now, to diagnose Alzheimer's, there are comportamental and biochemical strategies, biochemical analysis. And the idea is to try different techniques or more and more techniques which allow an earlier detection of Alzheimer's disease and also easier detection if we want. Relatively recently, it was demonstrated that an accurate blood test of Alzheimer's disease could streamline the diagnostic workup and treatment of AD. So in this case, they were looking of biochemical components in the blood of the patients with Alzheimer's disease. But there is also an alteration of the red blood cell by the beta amyloid, which can be speculated. So what is beta amyloid and Alzheimer's disease? Involve interactions with red blood cells because the blood circulates through the brain. So we have the beta amyloid peptides can bind to red blood cell, altering their morphology and function, which may contribute to the disease by reducing oxygen delivery and increasing oxidative stress. These interactions can occur in circulation, and the resulting damaged or altered red blood cells and beta amyloid binding to them may serve as potential biomarkers for Alzheimer's disease. So the question we address is the piezo 1 activity influenced, and we showed that beta amyloid peptides alter the biomechanical properties of red blood cells, and we observed also that significantly higher IODA-1 induced calcium responses in red blood cells for Alzheimer's disease. You might ask, what is IODA-1? IODA-1 is a chemical reagent which induces the opening of the piezo 1. So ideally, it could be pharmaceutic element which can control or can drive the piezo 1 channel. But the mechanical effect on the cells, it is interesting to study, and we did this through the microaspiration. And the results are published in a recent paper. They are a result of a project led by University of Eastern Finland with partners from Sweden and Italy. and it is published in Alzheimer's Dementia Journal. Okay, so briefly what we observed. We observed that the activation probability for a given pressure in case of control and alpha beta pre-incubated samples are different. And we observed that the difference are significantly, and when the cells are treated with peptides, the pressure for activation is increased. Then here are the representations of the values, the distribution of the values of the activated pressure, so is increased, and of the mechanical properties, Young's modulus and viscosity, which are decreased, so are reduced by treatment. Well, we arrived to conclusions. Maybe I was a bit long. I apologize if it was too long. Red blood cell mechanics and piezo-1 activation provides the use of red blood cell as biomarkers for different diseases. So it's interesting point because red blood cells experiments with red blood cells are mechanical and not only mechanical, are relatively easy and accessible. And red blood cell bring with them many information, a lot of information on different disease. Microaspiration is a useful technique to investigate mechanical properties and piezo-1 activation in red blood cell. We have developed a setup and a technique which allows concurrently measurement of mechanical properties and piezo-1 activity, and this allows to demonstrate a strong correlation between mechanics and piezo-1 activity. We studied the changes in piezo-1 activity and cell mechanics in different contexts. We discussed about actin instability, but we studied also different oxidative agents, and finally, treating the red blood cell with beta amyloid, a protein fragment initiating Alzheimer's disease, to which red blood cells are exposed during the blood circulation in the brain. For the last application, we demonstrate a net effect on the piezo-1 activity when red blood cells are exposed to the amyloid, and this might represent an additional way in AD diagnosis. I want to thank to my collaborators from CNR, from the University of Trieste, from Azienda Sanitaria Universitaria Giuliano Isontina, so hospital in Trieste network, University of Eastern Finland, and also to Fluigent for their device. And thank you all, and happy red blood cells because they are very important.
Thank you, Dr. Dan, for this great talk and all the conclusions that you have. It was really insightful, all the work that you had throughout this presentation. So now it's the time we start like to have your questions, and we can have like a discussions and answer all the questions that we receive. So the first one, it's from Enrico Ferrari, and he's asking about if, am I right to assume that the Ca2 plus intake due to the RBC membrane rupture rather than piezo channel opening can be ruled out because the mechanical threshold for the latter is much lower? Did you have to control for any other possible routes of the entry of the Ca2 plus?
Okay, there are two questions. The first, yes, we can rule out the entrance, the influx of calcium into the cell for rupture. In our case, so we observe at a much lower pressure the intake of the calcium. So, and the rupture, by the way, in many cases, it does not appear even when we suck the cell completely, because the channel, the diameter of the pipette is 2 micron, and in a 2 micron, many cells succeeded to go through. The second is, did you have to control for any other possible route of entry of calcium? No, I don't know. Yes, I don't know other routes excited in our cases. Probably you refer to different channels, not mechanical channels. But no, we didn't check control for these possibilities.
The second question, do the mechanical properties of white cell membrane also behave similar, according to your opinion?
Excuse me, I am missing this.
Yeah. Do the mechanical properties of white cells membrane also behave similarly?
I don't know. I don't know. From what I read, because micropipette aspiration was used intensively by Hochmuth, for instance, in the past to study white blood cells mechanics. I don't remember the values of the mechanics, but I didn't see any result of research on piezo-1 activation. In any case, without being an expert in this type of cells, I guess that besides piezo-1, there are also other mechanosensitive channels, and it is difficult to distinguish between the activity of them. So you have to block one channel and to study the other channel, but this is not always possible chemically.
Another question, how was the control of the pressure implemented? Was it manually or computer controlled during the experiments?
Yeah, I know. Yeah, actually what we did in the lab, we developed a platform, software platform to control imaging, so fluorescence, bright field, micropipette aspiration, and synchronize them to be able to develop our experiments. And this was done by Catalin, the guy who asks the questions. Yeah, it was an important contribution as this. Yeah. Because at the beginning we could control only the separately, so the pressure device and imaging.
Another question, how does the micropipette aspiration technique compare with other methods, probably that you tested, like the atomic force microscopy or the optical tweezers, in terms of sensitivity and its applicability to RBC studies?
Well, it is a range of forces of pressure, I would say, because in our case, we applied the millibar pressures. With optical tweezers, you go lower, you go one order of magnitude at least lower. And with AFM as pressure, you might arrive to this, but you might damage the sample in the sense that the force range in AFM is nanonewton, but the tip can be very sharp and the pressure as a consequence can be very high. But if it is too sharp, you damage the sample. And it is also very, I would say, not very, but quite difficult to understand when you really open the channels. I mean, to see the fluorescence in the same time. In any case, for instance, I am trying to work with red blood cells and with optical tweezers because optical tweezers can induce the formation of the cells, which are reasonable. You expect to have the opening of the channels, but I don't know yet if it is the too few channels are open and I don't have the sensitivity in terms of fluorescence, or the forces are not enough. The deformation is not enough. As type, I mean, microfluidics, they work also well, but you have to do a device to apply it. I mean, those microfluidic channels where you force the cells to pass through small diameter
Yeah.
channels.
So with this, I would like to thank you again, Dr. Dan Cojoc, for your insightful talk today and the participations with us. And I would like to thank you all for your participation, and feel free to contact us if you have any questions regarding the setup, regarding the applications. So thank you all, and see you in the next webinars from Fluigent.
Thank you. Thank you.
Bye-bye.